Hello everybody, and welcome back to Creation Myths. Today's creation myth is something you may not have heard of before. It's continuous environmental tracking. Instead of jumping right to the creationist claims like I usually do, I'm going to start the intro section of this myth by talking about where this comes from in just a little bit of detail, because it's really interesting in that it comes from basically one single source, one person. And that person is Dr. Randy Guliuzza, who is the president of the Institute for Creation Research, ICR, one of the big three young Earth kind of science ministries, along with Answers in Genesis and Creation Ministries International. And if you search for continuous environmental tracking, you basically only find Dr. Guliuzza and ICR. Check it out. This is the first page of Google results for continuous environmental tracking in quotes. It's all ICR articles by the president of ICR. You come down here, this is something he's written. Over here, same author, continuous environmental tracking with Dr. David Rives and Dr. Randy Guliuzza. It's all one person, basically. And I think this idea has a lot more clout than it otherwise would, because instead of being like some random person just kind of on the internet, it's the president of one of the big three young Earth organizations that is promoting this idea. So I want to get that out there up front. This is continuous environmental tracking. It's kind of a fringe idea, but one of the big three organizations basically not just buys into it or accepts it, but promotes it as kind of their thing, because the guy behind it is the president of that organization. So here is the creationist claim. It's kind of a two-parter, right? The first part of this CET claim is that natural selection isn't real. So I'm going to show you some quotes from Dr. Guliuzza in a piece called Darwin's Sacred Imposter, How Natural Selection is Given Credit for Design in Nature. So in this piece, he wrote, "Nobody has ever seen a selection happen. The words natural and selection in no verifiable way accurately describe observable interactions between an organism and its environment." Later on, he says, instead of convincing people that selection is real but really weak, it would be wiser to show them that selection is not really real. So they're going all in here on the idea that natural selection as a mechanism for evolutionary change is fake. It is not a real mechanism that actually describes how things happen in the real world. So that's the first part of this claim. The second part is, what's the alternative? If not natural selection driving adaptations in a population, along with, mutations and other mechanisms, but if not natural selection being a key part of that process, then what is? The answer is continuous environmental tracking, or CET. Instead of an organism or population experiencing mutations and having different phenotypes correlate with different levels of fitness, and the genetic traits that are correlated with the highest levels of fitness becoming more frequent over time, that's natural selection, instead of that, what CET proposes is that organisms sense their environment and adapt as needed. So I'm going to show you some excerpts from an ICR piece by Dr. Guliuzza describing how this process works. I'm going to have links to all these different pieces in the description down below if you want to read the full pieces. So he says, start with an observation. In this case, as environments change, creatures are soon observed exhibiting suitable self-adjustments. It seems like creatures can track environmental changes. Later on, he says the opposing view, as in the opposing view to natural selection, recognizes God's designing agency and so conversely sees creatures as active problem-solving entities. I want to highlight that phrase, creatures as active problem-solving entities. He goes on to say creatures drive themselves through time as they detect, take on, and solve very challenging dynamic conditions. He's very explicit about describing this in engineering terms, which I haven't kind of emphasized that part, but you can see here spectacularly engineered. He's very explicit about this being in an engineering framework. He continues, these innate mechanisms, these tracking mechanisms, yield results that are regularly described as regulated, rapid, often repeatable, and at times even reversible, words that better fit the outcomes of engineered systems. So one thing I want to point out here is there's a lot of engineering framing in this argument. I'm not going to go into it now because it deserves a longer treatment than just a digression in a video on something else. But there's this idea called the Salem hypothesis that basically the weak version, which is the version that I think is accurate, the weak version of the Salem hypothesis states that when you find creationists that have science credentials, it tends to be engineering credentials. That is definitely a thing. And you can like Google that and find like lists of like prominent creationists, and there's a lot of engineers on that list. And that's exactly what's going on here. Now, I do want to talk about the Salem hypothesis in more detail at some point in another video. This is how continuous environmental tracking works. It's basically using engineering principles to say the organisms detect their environment, they figure out what they need to do, and then they respond to their environment. And that's how adaptations occur and propagate through populations, not the process of natural selection. So now let's talk about why creationists are wrong about CET. The first thing, the obvious problem, is that natural selection does actually work. Here's that quote again. The words natural and selection in no verifiable way accurately describe observable interactions between an organism and its environment. And that emphasis is in the original, except there is nothing about that sentence that is true. For example, antibiotic resistance, cancer outcompeting non-cancerous cells, finch beak size after drought in the Galapagos Islands, human adaptation to high altitude in the Tibetan Plateau, pesticide resistance in mosquitoes, guppy life history traits in high and low predation environments, Tasmanian devil resistance to devil-faced tumor disease, which on its own deserves a video because devil-faced tumor disease is a transmissible cancer and the dynamics are fascinating. Tuskless elephants, nylon-digesting bacteria, bird flu codon adaptation to human hosts, antagonist to coevolution between countless predator-prey pairs, HIV adapting to humans, lighter coats in barrier island mice, of course SARS coronavirus 2 becoming more transmissible over time. These are all directly observed, directly documented instances of natural selection. The truth is that natural selection works. It is observable, we can describe it mathematically, and we can make valid predictions. The starting point for this whole CET argument that natural selection doesn't work as a mechanism of evolution is complete nonsense. But it gets even better, because CET was disproven 80 years ago, in 1943, in a very famous experiment conducted by Salvador Luria and Max Delbruck called the Fluctuation Test. And I'm going to tell you about this experiment because it's one of my all-time favorite experiments. This experiment sought to answer one of the fundamental questions at the time about how evolution works. The question was, do populations of organisms detect and respond to their environment with specific targeted mutations that contribute to adaptation? Or do mutations occur spontaneously and maybe by chance members of a population will have adaptations that confer an advantage in the future? The way they tested this was by growing bacterial populations. So each of these circles represents bacteria. Bacteria divide by binary fission, so it's 1 becomes 2, becomes 4, 8, so on. So they have bacteria. The bacteria are just growing in their cultures. And they put the bacteria on a plate. So you get different colonies on the plate, and they expose those colonies of bacteria to bacteriophages, viruses that infect bacteria. So if the bacteria were susceptible to the bacteriophage, then that colony would be destroyed. If the bacteria were resistant to the bacteriophage, then they would survive, right? They would not be destroyed by the bacteriophage that they were exposed to. So what they did is they took these bacterial populations, they put them on the plate, they did this a whole bunch of times, and then they documented the frequency of resistance among the bacterial colonies from their culture that they've grown. So you can see in this figure, they put it, this bottom row just represents the colonies on a plate, and they wanted to know, were the resistance mutations, the ones shown in red, were those induced by the virus, or were they pre-existing in the bacterial population before it was exposed to the virus? This experiment is beautiful because it makes very specific predictions in terms of what we should see with the distribution of resistant colonies if the mutations are induced versus if they are not induced. So here are those competing predictions. And before I go further, I want to credit a user called MadPrime on Wikipedia who made this figure. This is a beautiful illustration of exactly how this experiment worked. So here are the predictions. If you're inducing resistance mutations in these bacterial colonies in response to being infected by or exposed to by bacteriophages, then we should see an approximately equal number of resistant colonies on each plate. So if you do 4 trials here in this kind of simplified version, if you do 4 trials here, right, you should see about the same number of resistant colonies on each plate. If the mutations are not induced, we should see an extremely wide variation in the number of resistant colonies across the different plates, because in some cases, the mutations will have occurred late in the experiment, in that bacterial growth process, so you won't have very many colonies that have it. In other cases, it will have occurred earlier in the process, so you'll have a whole lot of bacteria that carry that resistance mutation. In some cases, it'll be somewhere in the middle, and in some cases, you won't have any resistance mutations at all. So if the mutations are not induced, we should see very wide variation in the number of resistant colonies across the different trials. This is a beautiful setup. I love this experiment. It's just very clear. If it's this explanation, here's what we'll see. If it's this explanation, here's what we'll see. It's great. So they did the experiment, and what did they find? The results were extreme variation in resistance, much more extreme in terms of the frequency of resistant colonies than what we see in this figure. This means that specific mutations conferring resistance were not induced by the presence of the bacteriophage. Those mutations instead occurred earlier in the bacterial growth part of the experiment, had nothing to do with exposure to the bacteriophage. That means 80 years later, we can say that continuous environmental tracking is wrong. It simply does not accurately describe how adaptations happen in biological populations. We do have another problem here. That additional problem is that individuals do not evolve. Populations evolve. This is one of Darwin's major insights way back in the 1800s. It's populations that change over generations, rather than individuals changing within their specific generations. But Dr. Guliuzza sure makes it sound like individuals are going to evolve under this CET framework. He writes, I'm gonna read this. It's a longer quote, but I'm gonna read it because I think it's relevant. We find that for most of the documented adaptations, creatures use elements that match well with those underlying the self-adjustable properties of human-designed tracking systems. These are 1. Input sensors to gather data on external conditions. 2. Internal programming specifying reference values and logic segments that compares input data to a reference and selects a suitable response. And 3. Output actuators to execute responses. That's how he describes this process of CET. Basically, it's input, and then there's some kind of logic, and then there's output. That's the response. That's the adaptation. Now, here's the thing. Is this supposed to be going on in a single generation? Because this is not how evolution works. We can just look in the world and see that you don't have individuals evolving new adaptations within single generations. That's just contrary, forget theory. This is contrary to just basic observations. You can go back to that long list of examples of natural selection that I gave you earlier. Look at the finch beaks, for example, right? After a drought. It's not that all the finches get larger beaks because they're just the big nuts available after the drought, right? It's not that their beaks get bigger in response to the different distribution of the food that's now present after the drought. It's that the ones that have big beaks tend to survive, so there are more offspring with big beaks in subsequent generations. So describing this as like an individual, consciously or not, detecting the environment and then responding to it with an adaptation, that's just flies in the face of like just what we see out in the world. But the alternative is no better. Because the alternative would be you split it across 2 generations. You have a parent generation that detects the environment and does the logic, but then somehow passes that information to the offspring who do the output part of it, who actually express the adaptation. In order for that to happen, you'd have to have some heritable change occur in direct response to the environment. And you may be thinking, gee, that sounds an awful lot like epigenetics, and you're not wrong. But if you read what they say about this, it's not about chemical modifications to DNA. They talk about mutations. They talk about the built-in capacity to change your DNA, right? So we're talking about specific mutations, changes to the sequence of the DNA molecule, not just biochemical modifications of it, that need to be induced and then heritable across generations. So really what this would need is you'd have to have the input sensors detect whatever the environmental challenge is, the logic to then cause a change in the germline, which then is expressed in the subsequent generation. That's not how this works. You can look for that mechanism. That's not how this works. I mean, it seems silly to just point it out like that, but it doesn't work like that. If that was how heritability and evolution worked, we would know it, because we would see that that's exactly what happens. We'd go, oh, look, these germline cells have different genotypes compared to the somatic cells elsewhere in the body. And when you express those new genotypes, you get this new adaptation in the next generation, right? We would have figured that out once we have genome sequencing. That's not what we see. That's not how it works. So you could do it in a single generation, but that's contrary to everything we observe in nature. You could split it across 2 generations. There's no mechanism for that. The mechanism that it would be doesn't exist. So this doesn't work no matter which way you do it. And here is a bonus for you. Creationists disagree with ICR and Dr. Guliuzza. Almost all creationists accept some degree of natural selection. So I'll just give you some examples. Here is Dr. Nathaniel Jeanson writing for Answers Research Journal in 2013, Does Natural Selection Exist? A critique of Randy Guliuzza's claims. He writes in this piece at one point, over and over, Guliuzza denounces and demonizes natural selection, all the while failing to address the process. We can go and look at Dr. Rob Carter, writing for Creation Ministries International. Natural Selection in Paradise, Why Natural Selection is Part of the Creation Model. That's from 2020. He also did a thing in 2021 with Dr. Jonathan Sarfati. Natural selection is fact, and it's not evolution. It is evolution. I think I've talked about that before, but natural selection is very much evolution. But the thing I want to focus on here is Carter and Sarfati saying that natural selection is fact. And then one more thing that I'll point out. We'll go over to my friends at Discovery Institute. We'll look at Dr. Michael Behe, Senior Fellow, Center for Science and Culture. Behe's current research involves delineation of design and natural selection in protein structures. Again, he wouldn't call himself a creationist, but he's a creationist accepting that natural selection is a process of evolution. Now, I do want to point out, at some point, I might just do a creation myth on its own of like natural selection isn't real, which is kind of a fringe claim, but it's kind of related to this. So we'll see if that's something I end up doing in the future. In summary, creationists, or one creationist and the organization he is president of, claim that natural selection doesn't work, and the alternative, continuous environmental tracking, is what actually explains adaptations. It turns out natural selection does actually work as a mechanism of evolution, and CET was disproven by a famous experiment from 1943. So there is absolutely no validity to this claim of continuous environmental tracking. So where does that leave us? Well, continuous environmental tracking, that is a creation myth. Thank you for watching. Please like, subscribe, share, and don't get fooled.